Hello everyone, my name is LaFave, and today I'll be talking about the diversity in myosin isoforms found in rodent jaw muscles. Muscle is a hierarchical structure, and within the smallest unit of muscle, the sarcomere, is myosin, the force-producing motor protein that hydrolyzes ATP and drives muscle contraction. Myosin exists as several different isoforms or versions that relate to function. For example, long-distance runners utilize leg muscles with slower isoforms that are better for sustained contractions, while sprinters utilize leg muscles that have faster isoforms, better for explosive, quick movement. One isoform that is relatively less understood is masticatory myosin, or MHC-M. Masticatory myosin is found in the jaw muscles of vertebrates all along the evolutionary tree, including rodents, which are the focus of my research. What all these species have in common is that they require a strong bite for various different reasons. While it is understood that masticatory myosin is found in jaw-closing muscles, it is not well understood the properties of masticatory myosin or how it relates to the structure of the jaw. This leads to my hypothesis, which is that only jaw-closing muscles express masticatory myosin. To test this, I looked at the jaw muscles of sciuromorphs, which refers to a skull type that includes squirrels, chipmunks, and woodchucks. I used SDS-PAGE to look at the myosin isoform expression, where the bands you see here correspond to specific myosin isoforms. I looked at 4 jaw-closing muscles and 2 jaw-opening muscles for several different species of rodents, and I compared the intensity of the bands, which correspond to the amount of each isoform. What I've seen is that the gray squirrel does express masticatory myosin, seen here in purple, in its jaw-closing muscles, including muscles that were not studied before. Meanwhile, there is no masticatory myosin in the jaw-opening muscles. A chipmunk is very similar to the gray squirrel, while the woodchuck starts to show some major variation. However, it still does express masticatory myosin in the jaw closers. So we can conclude from this that only the jaw-closing muscles express masticatory myosin. But if we look at red squirrels, we can see that none of the muscles express masticatory myosin, meaning there is great diversity in the isoforms of the jaw-closing muscles. This is super interesting, as these species have the same skull type with very similar morphology. In conclusion, in order to explain this variation, experiments at other scales need to be performed, like whole muscle experiments looking at how big the mouth opens, to smaller scale genetic sequencing to see expression patterns of other muscle proteins, to experiments at the same scale like in vitro motility to look at the properties of these isoforms. Thank you for listening.